This is Vance again. This is Unit 12 Notes, Part 5. We're talking about the importance of bacteria. So there are a number of different things that bacteria are important for. Like I said earlier, they're important for decomposing, or the recycling of nutrients in the ecosystems. If it weren't for bacteria and fungi, then when something died, it would never rot. And those nutrients would actually be trapped in the body. And so they serve a very important function in our environment by recycling those nutrients, like making them available to be used again. Another really important thing that bacteria do is nitrogen fixation. They convert nitrogen gas to nitrates, which allows plants to incorporate nitrogen. Nitrogen is very important for lots of things we'll talk about more in just a minute. And they're also important for genetic engineering. We can now engineer bacteria to make products for us. For instance, various kinds of drugs or human proteins or human hormones like insulin and growth hormone and things like that can be made by bacteria much more quickly and easily and in larger volumes than by harvesting it from individuals. And then, of course, there's some bacteria that are pathogenic, disease-causing bacteria. That's the smallest category there, because that's we think about it the most, but it's really a very small category. Only about 2% of bacteria are pathogenic. So let's talk about some of these things. So, in bacteria that live in root nodules and in the soil are ones that are responsible for plants being able to incorporate nitrogen. As you know, our atmosphere is about 70% nitrogen. There's lots of nitrogen available, but most organisms on Earth are not able to use nitrogen directly from the air. It is unusable to them. There are only certain species of bacteria, like the Rhizobium species and the root nodules, but there are others that live in the soil as well, nitrifying bacteria and so forth, that are able to take the atmospheric nitrogen and convert it into usable forms like ammonia and nitrates and nitrites. The nitrates and nitrites can be used by, and ammonia sometimes, can be used by plants to incorporate nitrogen into their tissues. Nitrogen is essential for lots of things because that's an essential component. If you think back to the beginning of school, when we learned about the biomolecules, things like proteins and nucleic acids require nitrogen. And if you didn't have a steady source of nitrogen, it would be impossible for things to grow. Nitrogen is also the major component when you put fertilizer on your yard, for instance, in the springtime, and the grass gets really green. That's what the nitrogen is making it get really green, because now the plants are able to continue growing or start growing and make more chlorophyll to start photosynthesizing more so they can grow more. And again, the nitrogen is necessary for that. And so the bacteria help a great deal in that process. There are some bacteria that cause diseases. And so even though they're the smallest proportion of all the bacteria that there are, they're still very important to us because we're always exposed to various kinds of pathogenic bacteria. Most of the bacteria that cause diseases produce some kind of a poison or a toxin. And that's what causes the illness that you get when you get the bacterial infection. Exotoxins are proteins that the bacteria can secrete into their environment. If you've ever had food poisoning, for instance, that's usually due to an exotoxin that is released by the bacteria that are growing in the food. Endotoxins are parts of the outer membrane of gram-negative bacteria, and those are things that can make you really, really sick when you get an infection with one of those kinds of bacteria. And so we have various ways to treat those kinds of things. Staph aureus is an example that has an exotoxin that is secreted by cell. Staph aureus is one that can cause skin infections. You usually think about staph infections, but it can also cause food poisoning, some kinds of food poisoning. An endotoxin is part of the gram-negative membrane of Salmonella, which causes a diarrhea disease. And that's very unpleasant, but it's because of the endotoxin that's produced by the plasma membrane. And so different ones can do different things, but basically those are the two main kinds of toxins that are produced. How do we know what bacteria causes what disease? Well, for a long time we didn't know that, and back in the 1700s, early 1800s, even that recently, people thought a lot of times that diseases were caused by bad air or whatever. But in the mid to late 1800s, scientists more people became aware of microorganisms, and scientists started to realize that they were finding bacteria in various kinds of illness processes. And the problem was that they had to figure out a way to prove which bacteria caused which disease. And so a doctor named Robert Koch is the one that came up with a way to do this. He came up with a set of rules that we still use today called Koch's Postulates. And these are conditions used to establish that one particular bacterium is the cause of a particular disease. So first of all, you have to be able to find that bacterium in every case of the disease. Okay? You need to be able to isolate the bacterium from a person who has the disease or an animal that has the disease and grow it in pure culture. And then you have to show that that cultured bacterium causes the disease when it's transferred to a healthy subject. And then you should be able to isolate the bacterium again from the experimentally infected subject. Now, generally these days we don't really do that, go through all those processes, because it's well established that bacteria often cause diseases. But those are the ways that to actually show which one is involved with which disease. Oftentimes we don't give the disease to a human to figure out if it's a human disease. We'll give it to something like a mouse or something like that we use in lab experiments. But that's really important. Now, we can't use Koch's postulates, really, exactly as they are, to diagnose viral diseases, because one requirement for Koch's postulates is growing it in pure culture. And you can't do that with viruses because viruses are not living. You can grow viruses in a culture of living cells, but that's not exactly the same thing. So there are a set of postulates that are used for to diagnose viral conditions or viral diseases, accounting for that. But you have to think about the difference between bacteria and viruses here, because viruses are not alive, so you can't grow them in pure culture without other living cells. Antibiotics are something that we often use to fight bacterial infections. Okay, these are naturally occurring chemicals that are made by living things like fungi and other bacteria as well. And most of them work by interfering with the cell wall formation. If the bacteria can't make new cell walls, then it can't grow. And if it can't grow, it's going to eventually die. Examples of antibiotics would be things like penicillin, that's like ampicillin, amoxicillin, things like that. Tetracycline, azithromycin, which is what you get when you take a Z-Pack. And so these are various ones that can be used to fight various kinds of bacterial infections. Here, the antibiotics were first discovered back in the 1920s, almost 100 years ago now, by a scientist who accidentally left a petri dish with bacteria lying open on his lab bench, and it got infected by a fungus, and he saw a clear area around the fungus where the bacteria weren't growing. He was like, wonder what's there. And that was where they were first discovered back in the 1920s. They didn't become widely available until after World War II. They were used during World War II mostly to fight battlefield infections. And then after World War II, they became much more common to use in everyday medicine. This is why, and most of you have probably had an antibiotic at least once or a dozen times in your life. So when we're trying to figure out which antibiotic is going to work the best, we do something called an antibacterial or an antibiotic resistance test. This is a petri dish that's got bacteria. The brown stuff here is bacteria growing all over the plate, and we put these little individual discs on there, each of which is impregnated with or soaked in a certain concentration of a certain kind of antibiotic. And you put those on the bacteria. You plate the bacteria, and you put the discs on there, and then you put the plate with the discs and the bacteria in the incubator and let the bacteria grow. Where the bacteria are not growing, in other words, the clear areas are going to, that's what we call a zone of inhibition. Okay, and so you can see that some of this particular one looks like it works a whole lot better than this one or this one. This one looks pretty good, but it's still got some growing around the edges. Okay, this one is a nice clear a nice clear zone around it. This one would not be very effective at all for it. In fact, that one may be actually a control of just a distilled water disc. And so there is an activity that you will do where you measure the zone of inhibition to figure out which particular antibiotic is the bacteria are susceptible to, which one would be an effective one to use for that particular infection. Now, how do bacteria develop resistance to bacteria? Well, it usually happens by means of a mutation of some kind. Okay, and once a bacteria has a mutation that enables them to survive a particular antibiotic, they can actually exchange plasmids with other bacteria. And plasmid exchange often includes antibiotic resistance genes. Okay, and so this diagram kind of shows you how some of that works, basically. But there are antibiotic resistance is a bigger and bigger problem these days in fighting bacterial infections. There's one particular one called MRSA, which is methicillin-resistant Staphylococcus aureus, which is also called the superbug. Okay, it's resistant to a lot of the antibiotics, and especially some of the really, really strong ones that you would use to fight pretty much everything else. Methicillin is one of those. Okay, so MRSA is a really bad staph infection, and sometimes they can be difficult to treat, if not deadly. That's one of the reasons you have to be really careful in hospital situations, especially. Because you're more likely to have resistant forms that have been exposed to more and more bacteria that have resistance to different antibiotics. And so one thing that's really important in terms of medical treatment, like in a surgical suite, like you see here in the picture, is that everything has to be sterilized from the ceiling to the lights to the floors to the equipment. Everything has to be sterilized to prevent the transference of bacteria. Why is this important? Well, and why do we have a picture of the gym here? Well, here you have people working out in the gym and they're touching each other. And what if one of them has a staph infection that can be transferred by touch to another person? And that can cause major problems. Some of you may have had a staph infection on your skin. And most of the time it can be treated with antibiotics, but it can be pretty serious if you have someone that is resistant like MRSA is. So how do antibiotics work? Well, one way they work is by interfering with the cell wall formation, okay? And so realizing that cell walls are different in gram-negative and gram-positive bacteria, there are a lot of antibiotics that don't work on the gram-negative bacteria because that peptidoglycan wall is not exposed. A lot of times they work by interfering with the ribosomes that are involved in making the peptidoglycan and other molecules that are necessary. Here we have just another comparison of the cell walls of gram-positive and gram-negative bacteria. Gram-positive bacteria have a thick layer of peptidoglycan that is outside the cell membrane. And if they're unable to make the components of that, then they can't make new cell wall to protect themselves, and therefore they're going to not be able to grow. But the gram-negative bacteria, again, have a really thin layer of peptidoglycan in between an inner membrane and an outer membrane. And so they're much more difficult to treat for that particular thing. They have to have antibiotics that work in a different way to fight those kinds of infections. A lot of antibiotics target the ribosomes, okay? And so, again, if the ribosomes are not working, then they can't make new cell walls to replicate, and therefore they can't grow. And so that would be a way to fight against bacterial infections. So what are the differences? There are a lot of different terms that refer to these things, and we've talked about some of these things, and you've talked about them in your personal life as well in the last years, especially when we've been dealing with the pandemic. So we need to know the difference between antibiotics, antiseptics, disinfectants, and antibodies. All of those things are important in fighting infections. So antibiotics, again, are the products of living cells that can be used to fight bacterial infections. They do not work on viruses, they only work on bacteria. Antiseptics are chemical compounds that can be used on living surfaces to kill potential pathogens. So, for instance, when you go to the doctor's office and you get an injection, or if you go get a vaccine, what do they do before they do that? They actually clean your skin with alcohol. And alcohol is an antiseptic that can be used on a living surface to kill bacteria. That way, you don't have to worry about when you get the injection them also injecting bacteria into underneath the skin. Bacteria that might be harmless on the surface of your skin may not be harmless when they're on the inside. And so that's an important thing. That's why they're really careful about cleaning your skin before they do an injection. Disinfectants are chemical compounds that can be used on non-living surfaces to kill potential pathogens. So that's what we use to clean the tables with. That's what you would use to clean surfaces. If you were working in the surgical suite in the hospital, then you'd be using disinfectants to clean the surgical suite to make sure that it was killing potential pathogens that were on the surfaces there. And then lastly, antibodies are immune system proteins that can help neutralize foreign substances by binding to them. And we'll talk about antibodies some more when we talk about the immune system coming up in the next part of this unit.